Good morning everyone. Good morning. Good morning Karan, good morning Jahnavi. Good morning Sivaranjani, good morning Shivani. Okay, let's wait for a minute, let others also join. We'll start it off. Okay. Good morning Srikant, good morning Mauli. Okay, so friends, let's continue. If everyone quickly recall in the morning class what we have discussed, friends, in the yesterday morning class, we were discussing about the hormone auxin, right? Okay. In the 11:30 a.m. class, I have finished the auxin and we started the gibberellic acid also. And we were about to finish the gibberellic acid also, right? Once again, we will be continuing this particular gibberellic acid part 2 in the 11:30 a.m. class only. Okay. So now in this particular class, we will try to finish the hormone abscisic acid, hormone ethylene, as well as the hormone cytokinin. So, and the little left part we will be finishing at the 11:30 a.m. So by 11:30 a.m. class, all the 5 hormones will be completed, and evening class we will do the PYQ discussion. Okay. Okay. So friends, in the yesterday class, I told to all of you that what is the best way to remember the hormones, friends? That is comparing one hormone with the another hormone is the best way to remember the hormones. Okay, if everyone quickly recall, the moment we heard the name of a auxin, what is the statement? Good morning, good morning. What is the statement I have given to all of you to remember? Auxin is the rooting hormone. Good morning. And the moment we heard the name of a gibberellic acid, I told to all of you, friends, remember gibberellic acid is the hormone that plays a crucial role in the process of a seed germination. Similarly, the moment you heard the name of a abscisic acid, 2 points has to strike in your mind. What is that 2 points has to strike in your mind? Good morning, good morning. The first point is abscisic acid is the growth inhibitory hormone. This is the first point has to come in your mind. And the second point has to come in your mind, friends. Abscisic acid is the major stress hormone which is released in the plant body during the condition of a stress. Okay. And friends, I hope every one of you remember in the yesterday class I told you, everyone quickly answer my question very fast. Gibberellic acid is having how many number of carbons? What do we call the gibberellic acid? Do we call the gibberellic acid as the monoterpene or the diterpene, friends? I hope every one of you remember. We called gibberellic acid is the diterpene, having how many carbon, friends? 20 carbon. I want you people to remember. Similarly, friends, when you heard the name of a abscisic acid, every student keep it in your mind. Correct. Abscisic acid is what, friends? Abscisic acid is the good morning, good morning. Good morning, Kritika. Abscisic acid is the sesquiterpene. So, friends, sesquiterpene having how many carbon containing compound? 15 carbon containing compound. Friends, please don't get confused. Once again, I'll explain to all of you in case of, secondary metabolites. Keep it in your mind, friends. Monoterpene is the 10 carbon. Sesqui is 15 carbon, and friends, diterpene will be the 20 carbon containing compound. Never ever confused with this. Friends, understand. Good morning. So, friends, the discovery of the abscisic acid, it was really, it's a kind of a different type of discovery. Everyone understand my statements correctly. Three different scientists from the 3 different labs, from the 3 different places. Understand everyone correctly? Follow me, follow me. 3 different scientists from 3 different places and from the 3 different labs. They have isolated 3 inhibitors. Persons are different, labs are different, places are different, and the type of inhibitors they have isolated is also different. And friends, what are the three type of inhibitors that they have identified? One is called as Darmin, another one is called as abscisic 2, and the third one we can call it as inhibin. Friends, after few years, everyone follow my words, after few years it was really identified that what it was identified, it was identified that these three inhibitors, friends, structurally also as well as functionally also, they are exactly similar. Understand my words. The three inhibitors identified completely differently, but it was surprising to know that, it was surprising to understand that the three inhibitors, friends, both structurally also, functionally also similar. So based on this three name, there was a single name were derived, that is only ABA, which we also called as abscisic acid, which is the major plant hormone releasing in the plant body during the time of stress conditions. Once again, always try to compare one with other, friends. You can remember it very easily. So once again, every one of you listen my statements. Whenever we were reading the auxin, what we studied, friends, auxin is the hormone which is abundantly synthesized in the apical meristem. Correct na? In the apical meristem it will be abundantly found, and anyway we are going to study it. When we are studying the gibberellic acid, what we understand, friends, gibberellic acid is abundantly found in the seeds, right? Even it is also found a little bit in the roots. Similar, why I am comparing, friends, when you compare you can remember it easily. Similarly, where the abscisic acid will be synthesized, I want every student has to remember abscisic acid is said to be ubiquitous plant hormone. Friends, why do we call abscisic acid as the ubiquitous plant hormone? I hope every student remember, we all call bacteria are ubiquitous in nature. Friends, why do you say bacteria are ubiquitous in nature? Because friends, we know very well bacteria present everywhere. Correct na? Bacteria present at the acidic pH, basic pH, neutral pH, low temperature, high temperature, low pH, high pH, low salinity condition, high salinity condition. The bacteria present everywhere. That's why we can call bacteria are ubiquitous in nature. Similarly, friends, everyone understand. Continue. Why do we called abscisic acid is the ubiquitous in nature? We are calling abscisic acid as ubiquitous in nature, friends, because it is found in every part of the plant body. But every student has to remember a point that when I use a word, it is found in all parts of the plant body, doesn't mean that it is present in an extremely higher concentration. I already told to all of you, friends, if the hormone want to show a powerful action, the hormone should always present at the lower concentration only. Lower the concentration, higher the activity. Always remember, higher the concentration, lower the activity. That is the reason, friends, what we can say, abscisic acid as a ubiquitous plant hormone synthesized in all parts of the plant body, extremely low concentration. Friends, please don't get confused with this particular point at all. And remember everyone, abscisic acid is primarily synthesized in the vascular tissues like xylem and phloem, and transported to the targeting tissues. So I want every one of you understand, okay? Suppose example, abscisic acid is synthesized in the shoot. It is traveling to the root via phloem. Friends, don't get confused with this. Because abscisic acid found anywhere, everywhere. Whatever the abscisic acid found in the root, it is traveling to the shoot. How it will travel to the shoot? Via xylem. Whether it is the shoot, whether it is the root, the presence of abscisic acid, friends, obviously in the very low concentration. Don't get confused with this. And once again, the asked part B question in the exam, naturally occurring abscisic acid found in which form? Okay, every student answer my question very fast. There are many type of natural oxygens that we have studied in the yesterday class. Among all, which is the most important natural form of oxygen? Every student fast. Which is the most important and natural form of oxygen? Everyone. The most important and natural form of oxygen is always IAA. That is what we discussed. So friends, keep it in your mind. What is the most naturally occurring abscisic acid in the plant body? Can anyone tell me what is the most naturally occurring abscisic acid in the plant body? What is this? Anyone? So I want every student has to remember the most naturally occurring abscisic acid in the plant body. Friends, we will be calling it as the zeaxanthin. Friends, please don't forget. Zeaxanthin, like however oxygen is the most natural form in the plant body, similarly zeaxanthin is the most natural form of the abscisic acid in the plant body. Friends, the point you people has to remember that abscisic acid can exhibit in the cis form also. Friends, abscisic acid can exhibit in the trans form also. Correct na? So but friends, the point I want you people has to remember for the exam point of view is, okay, okay. So friends, try to understand this particular point. Abscisic acid can be exhibited in the cis form also as well as the trans form also. But the point what every one of you has to remember for the exam is that abscisic acid, which is the natural form, mostly it will be occurring in the cis form only. So what do you mean? So does it not exist in the transform? It will be also existing in the transform, friends, but most of the time the transform of the abscisic acid will be inactive, whereas only friends the cis form is extremely active. This is the question they have asked for the part B. Which form of the naturally occurring abscisic acid is the most active form? Always keep it in your mind, friends, which will be the cis form. Please don't get confused. Okay, now every student quickly answer my question. What is the name of the precursor that is involved in the oxygen biosynthesis? Hope every one of you remember name of the precursor that is involved in the oxygen biosynthesis is what, friends? Tryptophan. Always compare, friends. When you compare, you can remember very easily. So why I am saying here, now what is the major precursor that is involved in the biosynthesis of abscisic acid? Once again I will ask you, friends, make sure that you will remember it. That is IPP. What is that, friends? IPP. Once again I am explaining, while explaining the third hormone I will ask everything. What is IPP, friends? Isopentenyl pyrophosphate. Can anyone tell me, friends, isopentenyl pyrophosphate is how many carbon containing compound, friends? Every student keep it in your mind. Isopentenyl pyrophosphate is the 5 carbon containing compound. From this IPP pathway only, friends, we can synthesize the abscisic acid. I hope every student remember in the yesterday class that tryptophan undergoing what kind of a pathway, friends, in order to synthesize the oxygen. Hope every one of you remember tryptophan undergoing, friends, IPA pathway. It is undergoing the IPA pathway, and finally, friends, it is going to give rise to the indole acetic acid. So the point what you have to remember here, similarly, friends, the IPP undergoing which pathway, friends? You have to remember the name of the pathways which you can expect mostly in the part B questions. The IPP undergoing a pathway called as, friends, mevalonic acid pathway. It undergo the mevalonic acid pathway, then it is going to give rise to what, friends? Abscisic acid. Please don't get confused with this particular point at all. And friends, this abscisic acid can be transported via both xylem also, friends, as well as it can be transported via as phloem also. Now all the students look into the diagram. Abscisic acid is start synthesized from the precursors called as IPP. Friends, from the IPP, everyone understand my points. From the IPP, it is going to give rise to the zeaxanthin, vialoxanthin, neoxanthin, xanthozole, ABA aldehydes. These are all, what do we call it, friends? You people no need to remember, don't worry at all. These are all called as intermediates. These are all called as intermediates which are involved in the production of the abscisic acid. So friends, the point is the IPP, by giving a lot of intermediates, finally it can synthesize this particular abscisic acid, and this pathway only we can call it as the mevalonic acid pathway. Friends, the moment abscisic acid has been synthesized, what is happening to this abscisic acid, friends? This abscisic acid is transported via xylem as well as phloem to all parts of the plant body. Don't get confused with that. So in a simple manner, remember, naturally occurring abscisic acid is the zeaxanthin. The precursor involved is the IPP. Once the production is over, it will be transporting via xylem also as well as the phloem also. Okay, now we understand what is abscisic acid. We understand how it is getting transported. We understand what is the precursor as well. Now for the exam point of view, what is the most important? I told to all of you, for the exam point of view, most important one is the functions, then friends followed by the signaling pathways. These two are extremely important for the exam. Okay, if you get the question from the signaling, friends, 90% of the chances are there. Signaling, you can get the part C question only. Even though you get the functions, when they compare with the other, when they give along with the other hormone, even functions also, especially in the matching column, you will get in the part C only. Okay, I will explain one by one points very elaboratively, friends, without confusion. Follow me. Abscisic acid, what the first point, it is inducing the stomatal closure. I have told you, friends, I have used a statement that abscisic acid is a stress hormone. Friends, every student remember my words. I am using the word stress hormone doesn't mean that abscisic acid is released all the time in the plant body. No, when the condition is stress, understand my words, when the condition is stress, during this particular stress condition, the level of abscisic acid will be rising. If there is a no stress condition, abscisic acid is not at all active in the plant body. See friends, remember my words, not only abscisic acid, any hormone in the plant body never be active until it is required. Remember, any hormone in the plant body never be active until it is required. Similarly, friends, abscisic acid is also going to rise its level when the condition is the stress. All the students follow my words. What the abscisic acid is doing, friends? It is inducing the stomatal closure to prevent the water loss by the transpiration. This point we are going to read very briefly in the next slide along with the diagram. Anyway, you all people know that during the transpiration time, what will happen, friends? The stomata will be opened, is it? So these are all the two guard cells, and between the two guard cells there is a presence of the stomata. Stomata will be opened and there is a lot of water loss occur. You know, right? You know very well, if abundant amount of the water goes out of the plant body, what happens, friends? The plant may die, right? So that time what has to be happen? This particular stomata has to be closed. Now that opening and closing of the stomata mediated by the abscisic acid, how it will happen, we will study in the next slide. And the second very important point, friends, remember my words, promote seed dormancy. Friends, so many confusing statements are there. Okay, follow correctly, and most important, remember it correctly. Remembering correctly is very important. When you remember correctly, you never confused in the options. Promoting the seed dormancy. Friends, what is dormancy? Dormant is the period where there is no fertilization. Follow my words. Dormant is the period where there is no fertilization. Friends, whenever there is no fertilization, What do you have to remember? There is a no seed germination. Whenever there is a no fertilization, there is a no seed germination. So then the plant enter into what condition? Very adverse condition like drying, desiccation. Friends, what is desiccation? Remember my words. Desiccation is the condition where there is absolutely no water content in the plant body. Now remember my words. Abscisic acid is doing what? Abscisic acid is promoting the seed dormancy, which means it is not allowing the fertilization to take place. It is not allowing the seed germination takes place. So it is promoting the seed dormancy. But friends, understand my words once again. Gibberellic acid, what it will do? It will just do the opposite function. What do you mean by that, friends? Understand my words very correctly. Gibberellic acid, friends, it is going to inhibit. What it is going to inhibit, friends? It is going to inhibit the seed sorry. 1 second. It is inhibiting the seed dormancy. So friends, once it will inhibit the seed dormancy, tell me what is going to happen? The moment it inhibits the seed dormancy, friends, there will be a fertilization. There will be a seed germination. In one line, I can tell you, abscisic acid is inhibiting the fertilization, abscisic acid is inhibiting the seed germination. Whereas gibberellic acid is stimulating the fertilization, gibberellic acid is stimulating the seed germination. So now what is the statement I want to convey finally? The statement I want to convey finally, abscisic acid and gibberellic acid both are acting antagonistic to each other. Both are acting antagonistic to each other with respect to the seed dormancy. Is it or not? Whatever the functions that are performed by the abscisic acid, the same functions just performed oppositely by the gibberellic acid. Is it or not? That's why we can say that both are antagonistic to each other. So suppose example in a given matching question where most of the students will get super confused. In a given matching question, they have given a statement saying that promote seed dormancy and inhibit seed dormancy, and they have given both hormones, abscisic acid also, gibberellic acid also. You people has to choose it very clearly without confusion. Abscisic acid is always promote the seed dormancy, gibberellic acid is always inhibit the seed dormancy. Never ever confuse with the points. Now the third point, this is also very confusing point. The third point, everyone pay attention. It promote leaf senescence independently of ethylene. Now I am explaining you very correctly. Follow me without confusion. First of all, friends, what is leaf senescence? Senescence means, friends, aging. Correct na? Aging, becoming older. Becoming older. Question is very clear. Which hormone is involved in the leaf senescence? Friends, don't get confused with my words at all. Which hormone is involved in the leaf senescence? Originally, friends, ethylene is the hormone which will promote the leaf senescence. Every student follow my words. Do not confuse. Ethylene is the one which will always help the leaf to become older. Then, friends, what about the cytokinin? When we are studying the cytokinin, you people get a clear-cut idea. You know, friends, what the cytokinin will do? Cytokinin delay cytokinin delay the leaf senescence, which means it never ever allow the leaf to become older. Now, ethylene is promoting the senescence, cytokinin is delaying the senescence. Then why we are talking in the abscisic acid? Remember my statements correctly. Abscisic acid is also promoting the leaf senescence, but I have written a line very clearly, independently of ethylene, which means, so not in all the plants, friends, especially abscisic acid will do the process of a leaf senescence or the aging only in the citrus plants, that is independently on its own. But In the exam, if they ask you a question, which is the major plant hormone involved in the leaf senescence, undoubtedly you have to choose the option of ethylene only. In the exam, if they ask you a question, which is the major plant hormone delay the leaf senescence, undoubtedly you have to choose the answer cytokinin only. Do not get confused with that. Now, the fourth one, very important. All points are very important actually. Follow me. Promotes root growth and inhibit the shoot growth at low water potential. By that it can inhibit the germination, it inhibit the seed growth. I already told you, right? Abscisic acid is promoting the seed, promoting the dormancy. As a result, it inhibit the germination, inhibit the seed growth. Look into the fourth point. It promotes root growth and inhibits the shoot growth. So friends, this is the condition. These are all the roots. Now every student understand. What is the condition? The condition is in the soil, the water concentration is extremely low. All the students follow my words. In the soil, the water concentration is extremely low. And you know very well, when the water concentration is low, first who requires more water? Undoubtedly root requires more water. Root will absorb the more water, then only friends rest of the plant body can absorb the more water. During that time, what the abscisic acid is doing, it is inhibiting the shoot growth. It will inhibit the shoot growth. As a result, what is happening, you know friends, understand my words. The root can be penetrated into the deep of the soil. Okay, friends, understand, the roots can be penetrated into the deep of the soil. Once the root can be penetrated into the deep of the soil, what happen? It will absorb more amount of the water for its anchorage, as I mean to say, it will absorb the more amount of water for its growth and development during the low water condition. So suppose example, if the shoot is active, shoot also starts absorbing the water from the xylem via root. Then what happens? Root is unable to get adequate amount of water. So always keep it in your mind, when the water content is very low in the plant body, immediately shoot growth can be inhibited by the abscisic acid and root growth can be enhanced. Root growth may be promoted by the abscisic acid. By that, root can enter into the depth of the plant, depth of the water or depth of the soil. They will absorb abundant amount of water. Don't confuse with that. All the functions you people has to read 2 to 3 times. By that you can remember easily. Next one, it promotes desiccation tolerance. Friends, every student follow my words. What is desiccation? Friends, desiccation is the condition absolute drying. Okay, you know what do you mean by the word? Don't confuse, every student follow me. Desiccation means, friends, the plant is about to reach zero water level condition in the plant. I want every one of you follow my words correctly. What I am saying, desiccation is the condition where it is an extreme drying, absolute drying, where the plant is going to reach a zero water level condition. You know, suppose you know that if the plant reaches to the zero water level condition, friends, undoubtedly plant will die. So what is happening, friends? Now this particular abscisic acid will stimulate an important protein. We call it as LIA protein. What is this? LIA protein. Don't get confused. L represents late, E represents embryogenesis. Late embryogenesis abundant protein. Friends, understand every one of you. What is the function of this late embryogenesis abundant protein? Understand. It will try to maintain little amount of the water in the drying conditions. As a result, the plant never ever enter into the zero water level. By that, the plant never die, even the condition is the drying. Once again, understand my points very correctly. Suppose the condition is desiccation, which is absolute drying, where the plant is about to reach zero water level condition. During this particular time, what is happening? The abscisic acid stimulate the production of a protein called as LEA. So you all people has to remember, the LEA protein is not at all releasing all the time in the plant body. The LEA protein is releasing in the plant body only during the drying conditions. As a result, once this protein got released, it will helpful in maintaining not too much amount of water, a very little amount of water. By that, the plant never reaches to the zero water level condition, and it will survive somehow during the drying condition. The LEA protein can be stimulated by the abscisic acid. So friends, these are all the major functions performed by the abscisic acid. Now let's talk about the first function very elaboratively. That is, induces the stomata closure to prevent the water loss by the transpiration. Everyone follow me correctly. Okay. First of all, I want every one of you understand the concept correctly. Here we are talking that abscisic acid plays a key role in the closure of the stomata. First of all, we all people has to understand how the how the guard stomata will be opened, how the stomata will be closed. That is the point I want every one of you has to understand. Please look into the diagram very correctly without confusion. These are all what cells, friends? Guard cells. Yes or no? These are all said to be guard cells, right? So friends, whenever abundant amount of ions, especially what ions, friends? Every one of you follow my words correctly. Whenever abundant amount of the potassium, abundant amount of the chlorine and water molecules. Follow my words correctly, friends. When abundant amount of potassium, chlorine, water enter into the guard cell, friends, what is happening? The guard cell will become turgid. Yes or no? More amount of the ions entered into the guard cell. As a result, what happen? Guard cell will increase its turgor pressure. So friends, because of the increasing its turgor pressure, what is happening, friends? The stomata is getting opened. Now tell me opposite. When the stomata will be closed, friends, so you can look into this. When the potassium comes out, when the chlorine comes out, when the water comes out, what happened? The cell, instead of becoming the turgid, what it will become, friends? It will become flaccid. When more ions are present inside, friends, we can call it as the turgid cell. When all the ions come out, the guard cell will become completely flaccid. And you know very well, the flaccid cell will obviously closes the stomata. Very, very clear, simple. Now you all people has to get a valid doubt. What is the doubt you have to get? Sir, you said that the potassium, chlorine, water, once they enter inside, stomata is opening. Once they come out, stomata is closing. Now what is the role of abscisic acid? That is a valid point, you have to get it. Now all the students look into the picture very carefully. This is the very, very unique pathway and so much confused pathway. Okay, I want every student has to understand crystal clearly this pathway. Okay, it's very important. See, this is what, friends? A guard cell. This is also what? Guard cell. So friends, whether the stomata has to be closed, remember my words, whether the stomata has to be closed or stomata has to be opened, everything is under the control of abscisic acid only. Why? Because, understand my words correctly, on the surface of the guard cell, there is a receptor is present. What, friends? On the surface of the guard cell, there is a presence of the abscisic acid receptor. To that, what will come and attaches? Abscisic acid will come and attaches. Now I am explaining to all of you elaboratively twice. Follow me correctly. What is the ultimate goal? Ultimate goal of the abscisic acid here is closure of the stomata. Okay, how? By two pathways. All the students follow my words. One pathway we can call as ROS pathway, which we can call reactive oxygen species pathway. Next one we can call, friends, IP3CADPR. I want every student remember the full forms also. You know, right, what is IP3? Inositol triphosphate. What is CADPR means? Cyclic ADP ribosylated pathway. Please try to remember the full forms also. If you can't remember the full forms, remember the short names without fail. Understand my words, understand me correctly. Abscisic acid involved in the closure of the stomata by two pathways. One we called ROS pathway, one we can called IP3CADPR pathway. Now everyone understand how abscisic acid is doing this. Let's talk about the first pathway, ROS pathway. Friends, our function is common. What is our function? Sending the potassium outside, sending the chlorine outside, sending the water outside. When we send all these out, what happens? The cell will become flaccid, then the stomata will become easily closed. Now let's see how. All the students follow me. Whenever abscisic acid will come and attaches to the abscisic acid receptor, all the students follow my words correctly. What abscisic acid will do? Immediately activating the ROS species. Now you have to ask, what are ROS species? What is ROS? Reactive oxygen species. Free radicals. Free radicals only we can call ROS species. Example, H2O2, O2 minus, OH minus. These are all basically called as what? ROS species. Follow me, friends, follow me correctly. You know, once the ROS species activated, they are doing majorly three functions. All the students follow me. They are majorly doing three functions. What is the first function? They will block. What they will be blocking? Understand, they will be blocking the potassium incoming channels. Follow my words correctly. They are blocking the potassium incoming channels and they are opening calcium incoming channels, because calcium is very important, friends. You know, right? Calcium is the ion which plays a key role in several physiological functions that are occurring inside the living cell. So that's why calcium is compulsory required. The moment calcium enter inside, potassium never comes inside. Moreover, all the students look into this. Whatever the potassium that is present in the cell, whatever the calcium that is, chlorine present in the cell, all comes out. So potassium is coming out, chlorine is coming out. Then what happened, friends? Obviously, the cell loses its turgidity. It will become completely flaccid. Now the flaccid cell obviously close their stomata. Simple ROS pathway. Now I am explaining you other pathway, IP3CADPR pathway. Now you all people has to get a valid doubt. The doubt you have to get is, does these two pathways occur simultaneously or does these two pathways occur step by step? That is the valid question you have to get. The answer is, both pathways will occur simultaneously. They never ever occur step by step. Remember my words. Understand, friends, ultimate goal of the two pathways is closure of the stomata. But both pathways occurring at the same time, because both pathways are under the control of the hormone abscisic acid only. Now let me tell you IP3 pathway. What the IP3 pathway? Look into this. Understand my words. Abscisic acid will come and attaches to the abscisic acid receptor. Friends, the moment abscisic acid will come and attaches to the abscisic acid receptor, what is happening? Abscisic acid is activating the PLC. We have studied in the cell signaling topic many times. What is PLC, friends? Phospholipase C. We studied many times. What the PLC is doing? PLC is breaking down the PIP2. I told you many times, friends. When we break down the PIP2, what do we get, friends? We get IP3 and a DAG molecule. But you remember IP3. IP3 and cyclic ADPR, you know what they will do? They will actually go and bring the calcium ions which are stored in the vacuole. And this is the question asked twice in the part B. What is the question they have asked? Which of the following secondary messenger is involved in bringing the calcium from the tonoplast? What is the tonoplast? Outer membrane of the vacuole. We called it as the tonoplast. Correct now, friends? Now understand. Now the calcium from the vacuole, where the calcium enter? Cytoplasm. And another point is what? So there will be, see, look into this. H plus pump got activated and all the proton goes out. Every student remember, when all the protons goes out also, what is happening? That will also prevent stomatal opening. It never allow the stomatal opening at all. So obviously what is happening? Look into the picture. Calcium entered into the cytosol. The moment calcium enter, what is happening, friends? Potassium is going out, chlorine is also going out. Then obviously what is happening? The cell will become completely flaccid. Then the flaccid cell will close the stomata. Okay. Now once again I am re-explaining. Whomever did not understand, follow me correctly. There are two pathways are there. One we can call ROS pathway, IP3CA-DPR pathway. Both pathways will occur simultaneously. Ultimate goal of both pathways is closure of the stomata only. So how this process is happening? Whenever abscisic acid command attaches to the abscisic acid receptor, what happen, friends? It will be activating the ROS species. When it activates the ROS species, what they will do? They open the calcium channels by the calcium come inside. They block the potassium incoming channels. They activate chlorine and potassium outgoing channels. As a result, potassium is going out. As a result, chlorine is also going out. Now all the ions gone out. As a result, the cell will become flaccid, stomata closed. Simple. On the other hand, what is happening when abscisic acid command attaches to the abscisic acid receptor? It is immediately activating the phospholipase C. It will produce the IP3. What IP3 will do? It will go and associated with the calcium channels which are present on the tonoplast. What is tonoplast? Outer membrane of the vacuole we can call it tonoplast. It will bring the calcium into the cytoplasm. The moment calcium comes into the picture, what happens? Potassium go out, chlorine go out, water molecule go out, even proton also go out. When the proton go out, you know, friends, what is happening actually? The pumping of the proton out also prevent the stomatal opening. As a result, friends, the stomata will get closed. This is how abscisic acid is involved in the closure of the stomata. Okay, I hope every one of you understand that without having any kind of a confusion. If you have confusion, read it once again and ask me. In the evening revision class, we will read it thoroughly, you know, in the practice session. Okay, is it clear, everyone? Quickly respond to me. The hormone abscisic acid fast. Is it clear, everyone? Okay, perfect. Okay, perfect. Okay, just 1 second. Yeah. Okay. Okay. So friends, let's try ethylene. Okay. Once again, friends, always try to compare one with other. The moment we are comparing, it is extremely easy. Okay. Ethylene. When we heard the name of a auxin, we remember that auxin is the rooting hormone, right? And when we heard the name of a gibberellic acid, gibberellic acid is involved in the seed germination, right? And friends, the moment you heard the name of a abscisic acid, just now we all people studied that abscisic acid is involved acting as the stress hormone, is it? Similarly, the moment you heard the name of a ethylene, 2 points has to strike in your mind. What's the first point has to strike in your mind? Ethylene is the major fruit ripening hormone. Ethylene is the major fruit ripening hormone, and the most important point, friends, ethylene is the hormone which is involved in the leaf senescence. What is a senescence, friends? I told you, aging. Okay, when the leaf become older, that is also exclusively mediated by the ethylene, right? And okay, so remember, you all people also know that we have studied in the yesterday class, all the hormones are derived from the different precursor, and the way they synthesize is different. But what is the speciality of a ethylene, friends? Ethylene is the only hormone which is gaseous in nature. Always compare, friends. Okay, quickly compare. Auxin derived from what precursor? Tryptophan. Gibberellic acid derived from which precursor, friends? Gibberellic acid is derived from the precursor called as GGPP. And just now we studied abscisic acid is derived from the precursor called as IPP. And friends, what about the ethylene? Friends, ethylene is the major precursor which is actually derived from the amino acid methionine. Never ever confused with this. You know very well what is methionine, the sulfur containing amino acid. Friends, once again I am telling you, remembering precursors is extremely important. Okay, so ethylene is required for the plant growth, development, and friends, remember, even ethylene is also required for the stress responses throughout the plant life cycle. So in a given question, if they ask you which of the following are the pair of stress hormones in the plant body, so you have to remember ABA also a stress hormone, ethylene is also a stress hormone. But if they ask you among these two, which is the most abundant, that will be the ABA. Okay. See, that's what, if there is an absence of the ABA, what will happen? That hormone ABA action can be mediated by the other hormone ethylene in case of example stress condition. If the level of abscisic acid is extremely low, obviously the plant will die. Obviously, see, if we are having a hormonal imbalance, what will happen? It depending on the severity of that particular defect, various complications will occur. Same way in the plant body also. Suppose example, if the abscisic acid is completely absent in the plant body, during the stress condition, complete water loss will occur in the plant body. Obviously, plant will die. The entire development will be stopped because they will be requiring during the stress conditions. Okay, so now listen. You know very well ethylene is what? A simple gaseous hormone which is present in the fruits as well as the vegetables. Once again, quickly compare. Quickly. Auxin. See, this is how I remember the concepts very faster when I am comparing. Auxin will be abundantly present in the apical meristem. Gibberellic acid I told you in the seeds as well as the roots also. And abscisic acid, just now we have studied that it is a ubiquitous plant hormone, I told you. Similarly, friends, the moment you heard the name of ethylene, all the students keep it in your mind. 75% to 85% of the ethylene, understand friends, 75% to 85% of the ethylene, it is present in all parts of the higher plants, including leaves, stems, roots, flowers, fruits, tubers, and seeds. Everywhere the ethylene is present 75 to 85%. But friends, keep it in your mind, where abundant amount of the ethylene will be found actually. All the students keep it in your mind. 100% of ethylene. What friends? 100% of the ethylene exclusively present in the senescing tissues and ripening fruits. Friends, please don't get confused. Okay, senescing tissues which are becoming older, ripening fruits which are becoming matured. By see, when the fruit matured, then only it will become ripened. Don't get confused with this particular point. Now let's talk the biosynthetic pathway. Okay, Is there any necessity to remember the biosynthetic pathway structures? Once again, I'm telling you, it's a waste of time. Okay, you don't need to remember at all. And once again, keep on remembering. What is the auxin pathway called as? IPA pathway. Okay, gibberellic acid, we haven't studied it, but I will tell you. Gibberellic acid pathway is a terpenoid pathway. What? ABSCISIC acid pathway, just now we studied na, we called it as mevalanoic acid pathway. And friends, what is this particular ethylene pathway called as Ang cycle, based on the name of the discoverer. You people no need to remember even a single structure also. And what you have to remember? Remember, you have to compulsory remember starting compound. You have to remember intermediate. You have to compulsory remember the end product. Okay. Now every student follow me. What is the starting compound? Of course, methionine. Methionine is the starting compound. What is our ultimate goal? From the methionine, I have to produce the ethylene. That is my ultimate goal. Now all the students follow me. No need to remember the structure, but understand the schematic presentation correctly. Okay. Methionine combined with what, friends? Methionine combined with the adenine. When methionine combined with the adenine, it is converted into what? Adomet. How? By an enzyme called as adomet synthetase. I have told to you many times, when you read the word synthetase, tase always uses ATP. Synthase never uses the ATP. This is a very simple point you have to remember. Synthase never uses the ATP. Synthetase always uses the ATP. So methionine combine with the adenine and it is going to give rise to the adomet, adenosyl methionine. By what enzyme, friends? By a enzyme called as adomet synthetase enzyme. Never ever confused with that particular point, friends. Now all the students follow me. Now this adomet, everyone, this is the point. Now this adenosyl methionine or adomet can be treated by an important enzyme. What do we call it, that enzyme? We call it as the ACC synthase enzyme. What enzyme, friends? ACC synthase enzyme. So understand my words. Adomet, upon treating with the ACC synthase enzyme, it is going to give rise to an intermediate product called as ACC. This is the question asked in the part B in the 2019 June paper. What is the question asked? The question asked is, which of the following intermediate is produced in the biosynthesis of a ethylene? What is the intermediate produced here? No need to remember the structure. We can call it ACC. Amino cyclopropyne carboxylic acid. What is that? Amino cyclopropyne carboxylic acid is the intermediate that is produced. And finally, friends, you know what is happening? This ACC can be treated by an ACC oxidase enzyme and undoubtedly it is going to give rise to what? Ethylene. Such a simple step it is. Once again, I will be explaining you the pathway. Every student follow me correctly. Methionine initially combined with adenine. In the presence of which enzyme? In the presence of enzyme called as adomet synthetase enzyme. By that, it is giving rise to the adomet, adenosyl methionine. The adenosyl methionine can be treated by an enzyme called as ACC synthase enzyme to give rise to an intermediate molecule called as ACC. The intermediate molecule, amino cyclopropyne carboxylic acid. Remember the name, not the structure. And this ACC can be treated by an ACC oxidase enzyme. Ultimately, it is going to give rise to the ethylene. So this is the question asked in the 2019 June, and if you look into the 2020 paper, okay, even the same question asked in the IIT JAM 2022 question paper also, the hormone which is involved in the wounding, the hormone which respond with the chilling injury, the hormone which is responding with the drought stress, the hormone which is released during the condition of flooding, all the condition the hormone ethylene is only releasing. You know, no, the plants which are releasing extremely lower temperature, which we can call chilling. See, like however our skin get damaged during extreme chilling condition, same way plant also get damaged, right? So that time obviously this ethylene is only getting released. I want you people to remember the exact functions performed by them and when they are releasing actually. Okay, friends, and once again, you look into the previous papers, more than 3 times the asked question, that is the triple response. Friends, remember my words. The moment you heard the name of a triple response, you should, only one hormone has to strike in your mind without having any second thought, that is the ethylene. I will tell you what is this a triple response, what exactly it is. Anyway, friends, name itself very clearly indicating, right? Triple response. What is triple response? It is performing 3 functions, or it is showing 3 responses, that is called triple response. Now the point every student has to remember for the exam point of view, does ethylene is showing the triple response in all the plants? No, that's the question. Ethylene, friends, it is altering the growth. Okay, so friends, remember, ethylene alters the growth pattern of a dark grown seedlings or the etiolated seedlings. Friends, don't get confused, okay? Dark grown is also correct, or etiolated is also correct. Don't get confused. Etiolated means dark grown only. Look into the picture. What exactly the triple response? Every student follow my words correctly. In order to understand the triple response, this is also a type of question. Which plant used as a model? We have used Arabidopsis thaliana. What we have used? Arabidopsis thaliana as the model plant. We have used Arabidopsis thaliana as the model plant in order to study the triple response. All the students follow my words. This is also seedling of Arabidopsis thaliana and this is also seedling of the Arabidopsis thaliana. This is not treated with ethylene and this is treated with ethylene. Now you only tell me, friends, what are the difference that you people observe? Tell me. I can tell you simple differences. See, whichever is not treated with the ethylene, see here the root got elongated. Okay, the root got elongated. But friends, every student look into this. Whichever treated with ethylene, what happens? There is a inhibition of root elongation. So that is one response. Inhibition of root elongation. Second one we can call exaggeration of the apical hook. Friends, you know what is exaggeration? Exaggeration means, friends, tightening. Exaggeration means what? Tightening. See, all the students look into this diagram very carefully, friends. Here you see this is called hook. This is called hook. You only look into the picture. Here hook is very free. Yes or no? Hook is very free. That's why root is elongated. You can see, friends, because of the hook, root is getting elongated. But you see here, look into this diagram. Hook is very tighten. You can see here very clearly. So what I want to conclude here, this particular ethylene causing exaggeration of the apical hook, nothing but tightening of the apical hook. Now look into this. Here, remember, here the entire hypocotyl, this is what hypocotyl. Here hypocotyl will be completely swollen, but here it never be swollen. Why? Because this presence of ethylene will inhibit and swelling of the hypocotyl. And second function is what? Inhibiting the root elongation. What is the third function? Exaggeration or a tightening of the apical hook. And once again, don't get confused. Ethylene is not at all showing the triple response in every single plant. It is showing only in the etiolated seedlings. Friends, in simple words, what I want to conclude is, here triple response means changing growth patterns. What friends? Changing the growth patterns only friends. We will be calling it as the triple response. Don't confuse, only in the dark seedlings. Okay. Remaining thing friends, we will be finish it in the 11:30 a.m. class. Okay. We have a less time. So what I am going to do is in the 11:30 a.m. class only, I will start from the gibberellic acid wherever we left. After gibberellic acid, I will continue the ethylene and I will finish the cytokinin, because in the evening class we have to do the revision. Okay. Is it clear everyone? Is it clear everyone? Please let me know if anyone having any kind of a doubts. I'll upload the PPT in the app. You can revise it once. Is it clear everyone? If you have any kind of a doubt till this particular slide, you ask me. Okay. And your job is, now you are having a time. Now your job, you are having a time before coming to the evening revision class. I am requesting every one of you at least spend 40-45 minutes, revise it once again. By that I will solve the PYQ. Out of 10 PYQs, if you are answering 6 PYQs, once again I am telling 70%, you are on a right track. Okay. Okay. I'll see you sharp 11:30 a.m. in the app. Okay. Thank you all. Thank you so much.